Good evening, everyone. Welcome you all to today's NPTEL session on RNA transcription. Okay, so today also we will be going to discuss about some questions on RNA transcription, and we'll talk about it and discuss your points and my description also. Okay, so let's wait for few more minutes, like 3 to 4 minutes, because people are also joining. So okay, we'll start from 6:10. Let's wait for 3 minutes only. Okay. And if you have any issue with my screen or my audio, then you can just tell me. Okay. That if you are not able to see my screen or if my voice is not audible to you, then you can tell me. Okay. Okay, so let's start then. So as I have already told you that we are going to discuss some assignments, some questions on RNA transcription. So let's first know what is RNA transcription. Last class we have discussed about central dogma of life and RNA as enzymes in the form of ribozymes in different types of ribozymes and their functions also. Okay, if you remember. So here in the right hand figure, you can see this is called central dogma of life. That means DNA is replicated itself to DNA. That is called replication process. Next is DNA transcribes to RNA. That is the transcription process. And then this RNA translated to protein and make the primary structure of protein. After that, this primary structure of protein leads to different type of secondary, tertiary and quaternary structures. Okay, so this process of passing information, flow of information from DNA to RNA to protein is called central dogma of life. Understood? So, but now we'll be discussing about RNA transcription only. Fine. So, transcription is the first step in gene expression and it involves copying a gene's DNA sequence to make the RNA molecule. Now comes into picture which type of RNA. Yes, DNA makes three types of RNA, that is mRNA, tRNA and rRNA in measure amount. There are several types of RNAs been discovered right now, but in measurely we can discuss three RNAs are present. And transcription is performed by enzymes called RNA polymerases, which link nucleotides to form a RNA strand. Okay, and they use DNA as a template molecule. Got it? And I guess this is quite basic, and everyone knows about this much basic. So let's move to our first question, that is, which RNA is abundant in human cell? Please try to answer this question. Write your answer A, B, C, or D in the chat box. I am giving you 2 minutes. Please try to answer this question. Write your answer in the chat box. Then we will discuss. Sorry, I just got disconnected. Okay, I am sharing my screen again. I guess my screen is visible to everyone. Okay, so let's start the discussion. I saw your answer. Some of you have said, sorry, many of you have said mRNA, and some of you have also said ribozyme. But the answer is, just a minute, rRNA. Okay, rRNA is the answer. Fine, people are joining. I need to admit them. So, okay, let's discuss about it. So here my question is, which RNA is abundant in human cell? Abundant का meaning है which RNA is present in a high amount of or high percentage. Okay, so mRNA, that is the messenger RNA, tRNA, transfer RNA, and rRNA is the ribosomal RNA. Ribozyme, we have already discussed. Ribozyme is the combination of RNA and protein. It is a ribonucleoprotein and it has important role in catalysis. Fine. So let's talk about the three major RNAs. First is mRNA. So what is the percentage of mRNA? Here you can see the percentage of mRNA is 5 to 10% only. You must have thought about this, like, yes, there are a lot of replication transcription processes are going on in our body and in every human, every living being. So there must be the reason that mRNA is highly in abundant. But no, mRNA has only 5 to 10% of total RNA, and it is required for transfer of information, that is gene to ribosome that encodes the amino acid sequences. Fine. These amino acid sequences are called this primary structure of protein. Fine. So next is your tRNA. It has 10 to 20 percentage of abundance of total RNA. So tRNA is present in only 10 to 20%, and it reads the code on mRNA, right? So if you remember it or recall it, because we have already discussed in our previous class, this tRNA has a secondary structure, right? This secondary structure, when we talk about tRNA, there must be this acceptor arm. Okay, I am writing A here. This is the acceptor arm, and there are four. This looks like a clover leaf, right? And this is the anticodon structure. So here it will be the mRNA. For example, this is the mRNA. Okay, this is mRNA. And here, for example, if there is any code is here, this is the anticodon. It reads, this is the structure of whole tRNA. It reads the codes of mRNA, three-letter code, and this acceptor arm receives this particular amino acid which codes for this three-letter code. Okay, this is the function of tRNA, that is a transfer RNA. It transfers the amino acids, right? Next is rRNA or ribosomal RNA. It has 60 to 80% abundance of total RNA. Understood? And it constituents of ribosome. So think about it. Ribosome percentage is large in every living being. If it is your prokaryotic organism or if it is your eukaryotic organism, each and every cell has number of ribosomes, and this number of ribosomes have made up of this rRNA, that is ribosomal RNA. Okay, so rRNA has the high abundance, that is 60 to 80%, while mRNA and tRNA has only 10 to 20% abundance of total RNA. Understood? Now, this question may arise that there are other RNAs like miRNAs, microRNA, small RNA, and other RNAs which have been already discovered in the in become a decade. So these RNAs have very less abundance, very less abundance. Okay, as their name suggests, they are small in number and their percentage of total RNA is also very less. Understood? Okay, I guess you all have been clear with this, like we have discussed it clearly. So let's move to our next question. Like replication, transcription also occurs bidirectionally. Is it true or false? I believe that you must have little bit of basic idea about replication, transcription. So then please try to answer this, then we will discuss. No issue. If you don't know the answer, then fine. We will be discussing it. Okay. Please write your answer in the chat box. I will see and discuss. Okay, so let's discuss it. Like all of you have answered false. That means this is your correct answer, right? Fine. So now let's see how is it happen then. See, transcription occurs unidirectionally and synthesize RNA chain from 5 days to 3 days direction, right? So this 5 days, 3 days, and so you can see this blue color template strand. This is DNA, okay? These two strands are strands of DNA, right? And this pink one is showing the RNA, okay? So here, this 3 days to 5 days strand acts as a template. 5 days to 3 days strand doesn't act as a template. Remember this. If you get any questions like this, then you can answer it promptly. So think about it. Always RNA synthesis happens in your 5 days to 3 days direction, and here your template strand is 3 days to 5 days direction. Fine. And here is your enzyme RNA polymerase, and you can see the direction of synthesis, that is 5 days to 3 days. Okay. So only one strand has been used, right? Because only 3 days to 5 days strand of DNA can act as a template. That's why only one DNA strand has been used, and it acts in a unidirectional way. But in case of DNA replication, both the strands act as template, and also the synthesis of DNA is the one direction, but there is Okazaki fragments also produce, and these Okazaki fragments become ligated later to produce a new DNA, right? So here we are talking about transcription only. So this transcription occurs in a unidirectional manner, not in a bidirectional manner. Fine. DNA replication occurs in bidirectional manner. Also, Okazaki fragments also you can see only in case of replication, not transcription. Fine. Okay. Here is your next question. Mark the statement which is incorrect about the transcription unit. Now let's move towards little bit of transcription mechanism. Okay. Now we understood that how transcription happen, in which direction, and what is the template strand. Now let's talk about transcription unit. It is transcribed segment of DNA. Eukaryotes have monocystonic transcription unit. Prokaryotes also have a monocystonic transcription unit. Immediate product of transcription is primary transcript. Which one it will be? See, it is said incorrect. Okay. Read the question again, then try to answer. Okay, then let's discuss it. Some of you have answered B, some of you have answered C. Okay, so I guess you got confused between monocystonic and polycystonic, and which one belongs to what. Okay, so fine, let's discuss about it. First, let's read the question again. Mark the statement which is incorrect about the transcription unit. It is transcribed segment of DNA. Yes, it is true. Obviously, DNA transcribes to RNA. So obviously it will be a transcribed segment of DNA. So it is correct. Next is eukaryotes have monocystonic transcription unit. So some of you have said it is wrong, but yes, it is correct. It is not wrong. Eukaryotes have monocystonic. Monocystonic means a single gene is transcribed at a time, at a single time. Prokaryotes have polycystonic transcription units. Okay? And immediate product of transcription is primary transcript. Let's talk about it more. Fine. So as I said that cystone means gene. Fine. Now this thing you understand. And for example, your transcription unit, this is your structural gene. And in case of eukaryote, a single gene is transcribed at a time. Not multiple genes come together and get transcribed and produce lot of transcript. It's wrong. It doesn't happen in case of your eukaryotic system. Understood? And here this transcription unit contains one promoter region, one terminator region, structural gene, that is the DNA template also, 5' to 3' strand, the coding strand, template strand. Coding strand means in 5 days to 3 days this RNA synthesis been happening. Okay, that's why it is called coding strand. And this is your template strand for RNA synthesis. Now you got it. What is your transcription unit? Understood? So this transcription unit carries information of one gene. It is called monocystonic, while a set of adjacent genes which transcribed as a unit called polycystonic. So in case of prokaryotic organism, we can see polycystonic form of transcription where multiple genes can be transcribed at a single time. But in case of eukaryotic, this transcription process is quite complex. There are three types of RNA polymerases and different types of enzymes required for it. And also after transcription, this post transcription modification also happens, including your RNA capping, tail formation. Okay, so this complex process cannot be done for a multiple gene at a time. Understood? That's why it is monocystonic, a single gene at a time. I guess now your doubt has been cleared that this prokaryote also have a monocystonic. No, prokaryotes have polycystonic transcription unit. Fine. So let's move to our next question. Name the site where upstream sequence is located. Where is this upstream sequence is located? We have just discussed about transcription unit. So I believe that you can answer this question. Prior to start point, after the start point, right border of DNA, or in the middle of DNA. Everyone please try to answer this question. If you read the question, it is quite a little bit easy and clear. So please try to answer this question. Okay, so let's discuss about it. So all of you have answered A, right? That means it's correct. Okay, prior to start point. So what is this start point called? This is basically called this transcript promoter region. Okay, from promoter region, this is the transcription initiation site. If you see this figure here, DNA, this is 5 days to 3 days strand, that is the non-template strand, and 3 days to 5 days is your template strand, and it has promoter and terminator. So before promoter, those genes are present are called upstream gene, and after terminator, those are present are called downstream of gene. Fine. So here this upstream sequences are must be present before the promoter or before or prior to the start point. So promoter region is the region where this RNA polymerase binds to the DNA template strand, and after that transcription process starts. Got it? So start point is the first base pair from which transcription starts. Upstream sequences are located prior to the start point, while sequences after the start point are called downstream of it. Got it? So this is basically a gene where it includes a promoter, terminator, and RNA coding sequence. Understood? Let's move to next question. Which of the following is true for a RNA polymerase activity? DNA dependent DNA synthesis, direct repair, DNA dependent RNA synthesis, or RNA dependent RNA synthesis. Okay, so let's discuss. All of you have answered C, so it is correct. Fine. DNA dependent RNA synthesis. We have discussed it many of times. So and this is your RNA strands moving in 5 days to 3 days direction. And now a new term comes into picture, that is transcription bubble. So what is this transcription bubble? Where this RNA polymerase binds, this is your RNA polymerase, this orange color thing is called RNA polymerase, and where this RNA polymerase bind to the template strand of DNA, it forms a bubble like form structure. Okay, so this is called transcription bubble, and this transcription bubble moves to in in a forward direction to continue the RNA synthesis or continue the transcription. So this is your active site, this is RNA polymerase, this is coding DNA, and this is new strand of RNA. Fine, and this is a template strand of DNA. Direction of polymerization is obviously 5 days to 3 days direction for RNA, right? So this transcription bubble is the new term we got to know. Okay, so here is your next question. Which of the following ensure stable binding of RNA polymerase at the promoter site? DNA photolase, sigma factor, DNA glycosylase, RECA. Now you understood that RNA polymerase is an essential enzyme for transcription process, and it needs to be bound to the promoter site for the starting of transcription. So which one of the following helps in this process of binding? Okay, so most of you have answered B, some of you have answered C. Okay, so here is your answer, that is sigma factor. Got it? So what is DNA photolase and DNA glycosylase? Can anyone tell me? Write in the chat box. DNA photolysase and DNA glycosylase. From this name, this ASEs, that means they are enzymes, right? They are enzymes for what? Which process? They help in which process? Very good. They help in DNA damage repair. Okay. So during the replication process or after that also, if this DNA is damaged of somehow because of UV light or anything else, then these enzymes help in the breaking of this. Specifically, if you remember, DNA photolysase, it requires this visible light and it repairs UV mediated damage. If because of UV radiation something damaged, then DNA photolysase helps in DNA repair. Understood? Next is DNA glycosylase. When there is base pair damage, like uracil base pair damage, then DNA glycosylase helps in that particular base pair repair. Understood? Next is REK. REK is required for recombinational damage repair. Understood? So this REK helps in case of prokaryotes. It is required for recombination mediated repair. Got it? So now let's talk about sigma factor. So sigma factor decreases the binding of core enzyme to non-specific DNA sequences and increases binding to the promoter. So when there is a promoter present in the transcription unit, in I discussed it, right? Transcription unit has promoter, terminator, and between these two there is the structural gene. So when this RNA polymerase binds to the promoter region, this sigma factor decreases the binding of core enzyme to non-specific DNA sequences. It won't allow the enzyme to bind any other site. Got it? So this is basically called sigma cycle. So if you see the picture here, promoter engagement, that means this promoter is present where sigma factor will bind, and this will allow this RNA polymerase to bind only to the promoter region, not to any other region. After that, produces your initiation process will be there, then elongation process will be there, then termination, and after this termination sigma factor removes out. And then if the uh, transcription begins again, then it will come to make continue its role. Okay, so this is called sigma cycle, where this sigma factor is required for making sure of this promoter region should be bound by RNA polymerase only, not any other sequence. Fine. Okay, so what is the work of sigma factor in transcription? Now we have already discussed it, so please try to answer this question, everyone. Okay, so all of you have answered it. So I think you understood what I said. That is transcription initiation. Okay, not elongation, not termination. For termination, there will be another factor called row factor. We'll be discussing it in a few minutes. Okay, sigma factor is only involved in transcription initiation, not in elongation. Some people get confused that if it is helpful in initiation process or elongation process. So remember it is initiation only, not elongation. The sigma factor is released after the core enzyme when the transcript reaches more than 8 to 9 nucleotide. That means if the elongation process starts, okay, if the elongation process starts, then the sigma factor removes from the promoter region, or what do you say, core enzyme. So remove from it and allow the RNA polymerase to continue its work by RNA synthesis. Okay, so there are several sigma factors already present. So please name the sigma factor which is used for promoter recognition. Sigma 32, sigma 70, sigma 60 or sigma 40. Which one is it? This type of questions you will get in your UGC NET exam B section. If you know about this UGC NET exam, then there is A section, B section and C section. So this type of questions come in this B section. Okay, let's see then. So some of you have said B, some of you have said A. Which one is it? Let's see. See, there are 7 different types of sigma factors have been reported in E. coli, but the most common is sigma 70. Why this is very common? Because if you can see here, these are the genes and these are the sigma factor. If you see here sigma 70, it is a it generally acts for this TATAAT sequence. That means you must have heard about it. There is a tata box, that is TATA is sequence is present at the promoter region, which helps the sigma factor to bind to it. Understood? So this tata box, if you must have heard about it, then this sigma 70 only recognizes this data box, not 32, because for 32 the sequence is different, for 54 it is different, 28H, these are all different sequences. But if you see about sigma 70, which is very common, it generally 16 to 18 base pair in separation, and it is specifically recognized by this data box region, TATA. So that's why we'll go for sigma 70 for promoter recognition. Okay. Okay. So let's move to next question. Which of the following is used to describe the time taken by RNA polymerase to leave the promoter? Promoter clearance time, abortive initiation, elongation factor, or mean time? Please try to answer this question. Okay, let's discuss then. As all of you have said A, so that means it is the correct answer. Promoter clearance time. So why it is needed that RNA polymerase should leave the promoter? Because in the starting site, that is the initiation process, this RNA polymerase should bind to the promoter region, or there will be, if this enzyme binds to any other region, then that will be wrong gene transformation will be happen, right? So after binding to the promoter region, there is a specific bonds been made. Okay, so for that, this bond should be broken down, and this RNA polymerase should move out of this promoter region to move towards your elongation process. And how this happens? Generally, if you must have heard about these words like scrunching, inchworming. Inchworming means this RNA polymerase just moves away from little bit of promoter region and walk through the DNA. Like if it is the DNA, for example, this is the template strand of DNA, right? This is your 3 days and 5 days region. Because template strand is 3 days to 5 days, and this is your promoter region. For example, this is your promoter region, and here your RNA polymerase bind. And now this RNA polymerase need to leave this promoter. Okay, and how this happen? By three different methods, including inchworming and your scrunching, okay, and transient exclusion process. There are three processes for that. So how this happens? So for that, this RNA polymerase moves on the template strand of DNA little bit like a worm, okay, like a worm, like this, move like a worm. Okay, this is called inchworming. What is transient exclusion? RNA polymerase moves little bit on the DNA template, then saw back that if it is correct or not. That is transient exclusion. Next is scrunching. Scrunching means it will be present here only, but it will attract the DNA towards it. It will pull the DNA towards it. That is called scrunching. So it is believed there are three different hypotheses how this promoter clearance happens. Understood? So let's talk about it more. So promoter clearance is the time taken by RNA polymerase core enzyme to leave the promoter. More the efficient promoter sequences, lesser would be the promoter clearance time. Got it? So this is your minimal initiation complex. After addition of ATP to ADP, active transcription complex have been made, and after that promoter clearance. So here this ATP to ADP, that means energy is required for this process. You can see here energy is required, and after that the C terminal domain phosphorylation happens, and promoter RNA polymerase leaves the promoter. Okay, so this is overall view of your promoter clearance, where elongation process, this is the RNA polymerase now moves away. Okay, so this is overall a discussion about promoter clearance. I guess everyone must have understood it. So let's move to our next question. How many base pairs of DNA is transcribed by RNA polymerase in one go? So here we are talking about elongation process, right? Earlier we have been discussing about initiation process. Then we just discussing about elongation process. Then we'll be discussing about termination process. Okay, I see most of you have answered A. So let's see. Your answer is D. 7 to 8 bases. RNA polymerase core enzyme moves along the template strand at uneven base, that is moves over 7 to 8 base pairs in one go. So most of you have answered 5 to 6. So it might be correct also. See, it is 5 to 6, 7 to 8. This is quite a little bit similar kind of. Okay, so don't worry about it. So sometimes in the this Google and everything, these numbers vary, so it is not 100% true that 7 to 8 will be 100% correct and 5 to 6 will be 100% correct. Okay, so these are the experimental done things. So it might be 5 to 6 or 5 to 8. This is the range. Okay, fine then. Let's move to next question. Which of the following transcription termination technique has RNA dependent ATPase activity? Now we have been discussing about initiation process, then elongation process. So let's talk about termination process. Intercalating agents, row dependent, row independent, or rifampicin. Please try to answer this question. Okay, then let's discuss. Most of you have answered B and A. Some of you have answered A also. So let's see. So first thing, intercalating agents. What is this intercalating agent? These are the agents which attacks the base pair, that is intercalation. This process is known as intercalation. That means it affects the DNA bases only and inside the base pair. That's why it is called intercalating agent. Here we are talking about transcription termination. So in this transcription termination, which has RNA dependent ATPase activity, that is your row dependent. I have already told you that the row factor is essential for transcription termination, And there are two processes of transcription termination, that is row dependent and row independent. This row independent process is also called intrinsic process. Okay, this rifampicin is an antibiotic. We'll be talking about it. Fine. Okay, let's see. So this row factor functions as a hexamer and has an RNA binding site and is responsible for the termination of transcription. Fine. So this is row dependent transcription termination. So if you see here, this is the elongation process, promoter region, RUT rut site, then stop site. Here it is the termination site. 5 days, this blue color one shows the nascent RNA or primary transcript. Fine. This is your RNA polymerase, this enzyme. When uh, if you can see here, this rut site is there. That means it has already been transcribed, right? Through the when this RNA polymerase moves through this rut site, that means it got transcribed. And here at the RUT rut site, your row factor binds here. Understood? So after this row factor binding, it allow this ATP to ADP, which is a energy dependent process, allows the out pulls out this nascent RNA from this transcription bubble or transcription unit. You can say it bubble also. Though that means here it is helping to moving out. That means it just pulls out this nascent RNA or primary transcript from the transcription unit. You can see here, right? At first it was here, now it is moving away. So, and now after the end, release of RNA polymerase and completed RNA chain. So this RNA chain has been removed also. Got it? So here this your row factor ATP dependent process helps in termination process. Okay, and just pulls out the primary transcript of RNA. Got it? Let's talk about it more. So this row dependent termination signaled by a sequence in the template strand of the DNA molecule, okay, which are 40 base pair in length and are inverted repeat or hyphenated. These signals recognized by a termination protein called Rho factor. Rho is an ATP dependent RNA simulated helicase which binds to the signals. Thus RNA polymerase cannot move further, so it dissociates from DNA that disrupts the nascent RNA DNA complex. So what is this RNA DNA complex? You can see here I have rounded it. So this is your RNA DNA complex. Here is your nascent RNA primary transcript and this is your template strand of DNA. So this RNA DNA complex release the nascent RNA or primary transcript. Understood? Why it is called nascent RNA? Because this is the primary form of RNA. After this, there must be some post-transcriptional modification which will lead to mature RNA. Understood? That's why it is called nascent RNA and mature RNA. So this nascent RNA has lot of introns and exons also. But when this post-transcription modification happens, your introns, that is the non-coding sequences, have been removed, only the exons become present there. Got it? So let's talk about row independent termination, how this happens. So in case of row independent termination, it is most independent terminators have two distinguishing features. What are these two features? Let's see. One is palindromic One is palindromic GC rich region. GC means guanine and cytosine rich region, which is followed by an AT rich region. Thus, RNA transcript of this palindrome is self-complementary sequences, permitting the formation of a hairpin structure. Okay, so let's see. Let's talk about this hairpin structure. So if you see here, just I was talking about this only. So at particular site of termination or particular site of where this structural gene ends, this has some sequences that is GC rich, that is guanine and cytosine. So these GC rich sequences, they form a hairpin structure. That means they form bonds between them. You can see here, right? This is your hairpin structure. So after the formation of hairpin, your transcription bubble cannot move forward. That means the transcription needs to be stopped. Okay, sorry. Next is your second feature is a highly conserved string of A residue, adenine residues in the template strand that are transcribed into uracil residues. The RNA transcript ends within or just after them. Fine. That means यहाँ पे for example here what happens? First thing your hairpin structure formation. Second thing is your highly conserved string of A residues. Okay, adenine residues. So for adenine there will be uracil for RNA. So if a long chain of uracil comes out, then at that point of time RNA transcription stops, or after this uracil come it will stop. See here. Okay, pre-RNA. If there are multiple sequences of adenine residues are already present, or if that leads to uracil sequences, then that just terminates the sequence process of RNA synthesis. And here also by hairpin synthesis, it terminates your RNA synthesis, or it is overall it is called intrinsic termination. That is row independent. Here we don't require any ATP dependent method of termination, or we don't require row factor here. But in case of row dependent termination, there must be one binding site. Here is your transcription bubble, then it just removes out this transcription nascent RNA from this transcription bubble. Got it? I guess it is clear for you everyone what is the difference between row dependent termination and row independent termination. Okay, for row dependent termination we require ATP, but intrinsic termination or row independent termination we don't require ATP. Fine. Okay, so here is your next question. Name the one intrinsic terminator of transcription. I have already answered it a lot of time. Still, here is the question, so try to answer it. Intrinsic terminator of transcription. Okay, so I see some of you have answered C and some of you have answered B. So your answer is okay. I'll wait for few minutes then. You can answer. Okay, fine, then let's discuss. So your answer is row independent. Okay, first try to understand the question. Name the one intrinsic terminator. Intrinsic means inside. Okay, which is the terminator, or which is the factor which is already present inside. No outside factor is required, so it will be row independent. Those who have answered C, row dependent, please try to be focused on this figure. Here you will see that in case of your intrinsic termination or row independent termination, you don't require any row factor. Got it? And here also you don't require ATP. ATP and row factor both are absent in case of intrinsic termination. Because of the base or the sequences, already this hairpin sequence forms. Why? Because of GC rich sequences where G forms a triple hydrogen bond with C. So because of this hairpin structure or because of this adenine residues, transcription termination happens without any intervention of ATP or row factor. Okay, that's why it is called intrinsic termination, because this is happening inside. It won't require anything from outside. Understood? And in case of row dependent termination, there is one row binding site. Okay, and this row binding site is responsible where this row factor will bind. Okay, this will bind here. After that, ATP becomes ADP, means ATP is required, energy is required for this process, and this row factor don't allow the RNA polymerase to move forward. It won't happen. RNA polymerase will be there for a few time. So ultimately what will happen? RNA polymerase will move away, and this nascent mRNA or nascent RNA will get pulled out by this row factor. Understood? So nascent RNA becomes pulled out by this row factor. So here we require ATP, here we require row factor. That's why it is not intrinsic, it is row dependent termination. But in case of intrinsic or row independent, we don't require anything. Fine? Okay. I guess it's clear for everyone now. We have discussed it for a longer time. Okay. Still, if you have any question regarding this row dependent or independent termination, then you can ask these questions. Okay, no problem. So row independent termination is also known as intrinsic terminators, and about 50% of E. coli genes have this type of terminators. Okay. Next one question is, mark the one which is not the transcription inhibitor in eukaryotes. Rifampicin, acridin dye, actinomycin D, or row factor. Not the transcription inhibitor in eukaryotes. Here, if you have knowledge about acridin dye, actinomycin D, rifampicin is an antibiotic. So if you know about this, then you can answer this question. Don't worry, if you know, then you can answer or just guess it. Okay? Try to answer. We will discuss about it. Everyone, please try to answer this question. Okay, then let's discuss. Rifampicin inhibits beta subunit of RNA polymerase II, while acridin orange and actinomycin D is used to block elongation. ठीक है? Then your answer is D, because rho factor is the transcription inhibitor in case of prokaryotes. But here it is asked, mark the one which is not the transcription inhibitor in eukaryotes. So for eukaryotes, these all three of them are the transcription inhibitors. Rho factor is the transcription inhibitor for prokaryote. Okay. Next is coupled transcription translation is not for increasing protein synthesis, facilitating transcription termination, RNA protection, prevent RNA secondary structure information. First, let me know where this coupled transcription translation happen. In prokaryotes or eukaryotes? Everyone, please try to answer this question. If you read the question again, then you can understand it. We have already discussed about these things. Okay, let's discuss it then. Fine. So coupled transcription translation, that means when transcription and translation happen simultaneously. That is not for increasing protein synthesis. So obviously coupled transcription and translation will increase your protein synthesis, right? Because if your translation been happening simultaneously. Facilitating transcription termination, no, it won't be helpful for facilitating transcription termination, because we have discussed in case of prokaryotes row factor is responsible, or if it is not row factor, then the intrinsic method of termination is already there. So coupled transcription translation don't help in any kind of termination. Okay, next is your RNA protection, it might be true. Next is your prevent RNA secondary structure formation, this is also true. So if your translation happens right after transcription, or it is happening simultaneously, then it won't allow any time to form any secondary structure. Also the information of mRNA is highly stored, it is not damaged. If you give some time to it, then it might have some problem with that. Okay, but this is the thing in case of prokaryotes, not in case of eukaryotes. So here is this figure, you can see here, this is RNA polymerase enzyme, this is the template strand of DNA, mRNA, and this is 30S ribosome, enhanced recruitment of ribosome 30A subunit. So right after this transcription happens, so here you can see the chain of RNA have been increasing in the elongation process. So, and after this promoter region have been passed, it attaches to the mRNA. Translation commences along with 50A subunit, and translation stimulate transcription elongation. Okay, this is the thing happening in case of prokaryotes, not in case of eukaryotes. Understood? So this here translation and transcription occurs simultaneously. Understood? Because in case of prokaryotes, everything happens inside the cytoplasm. But if you talk about eukaryotes, this replication happens inside your nucleus. And after that, this mRNA needs to move out of this nucleus to cytoplasm, where this translation process happen, because ribosomes are present only there. Got it? Ribosomes are present in the cytoplasm, and their different types of amino acids and other enzymes are also present, which will be helpful in your translation process. Got it? And other thing also, there must be post transcriptional modification in case of eukaryotic, but not in case of prokaryotic. Because if you just translate the mRNA which just comes out from the transcription, then it will just take out all the introns also, non-coding genes also. So we don't require non-coding gene, we require only the coding genes. That's why after this post transcriptional modification only translation happens in case of eukaryotes, because the length of gene is also longer. Okay, so I guess it's clear for everyone. Let's move to next question. This is the last question of today's session, so please try to answer. mRNA capping do not ensure protection, translation, splicing, alternative splicing. So if we talk about post transcriptional modification, there is two things happen, that is capping and telling. Everyone knows that, right? Capping and telling, everything is detailedly described in your lecture videos, so go for the lecture videos if you don't understand any concept. Okay, so I got answers D. Some of you have said B also. So D is your answer. Alternative splicing. Okay. So this alternative splicing mechanism, the mRNA cap is highly methylated modification of 5' end of RNA polymerase to transcribed RNA. So when this 5' end is highly methylated, that is methylation cap is present here. If you see over here in the figure, okay, methylation cap. It protects RNA from degradation, recruits complexes involved in RNA processing, export and translation initiation, and marks cellular mRNA as self to avoid recognition by the innate immune system. So now you understand why this capping is so important for mRNA, because it is just add the tag as self. Otherwise, your immune system will going to attack on that mRNA, that nascent mRNA, and it's going to die. So here, this 5' end of RNA polymerase 2 transcribed RNA. It has this methylated cap. Next, here 7-methylated guanosine triphosphate. Okay. Also, this is overall called as the cap. 7-methyl guanosine tri. Also, there is polyadenylyl chain also there, which also helps the mRNA to get protect the mRNA from degradation. Okay, this capping and tailing, though both of these mechanism are important. Understood? So today we have discussed about RNA transcription in detail, like including initiation process, elongation process, and termination process. There are different factors, sigma factors, rho factors are responsible for initiation and termination process, and all of these there are different types of enzymes also play crucial role in total method of transcription. Fine. So we have discussed about a little bit. I think it will help you to do your assignments for this week 3 quiz assignment. And if you have any question regarding today's session, then you can ask me in the chat box. So any of you have any question? If you have any question regarding today's session, then you can ask me. Okay, then
Ma'am,
why
can you
don't,
explain
yes,
CTD phosphorylation?
yes, yes.
CTD phosphorylation, ma'am.
C terminal domain phosphorylation. Yeah, I think we don't have any question regarding that, but I'll explain to you. 15th question. Okay, fine. So here it is called mRNA capping do not ensure protection, translation, splicing, alternative splicing. mRNA capping do not. So here for do not, it should be like, yes, it ensures protection, right? So it will be out from fourth option. Next is translation. It do not ensure translation. Like if this mRNA capping is there, it helps transcription. Like the translation machinery will understand that this mRNA here only exons are present, no introns are there. So now this mRNA is called mature mRNA, and that then it will be just translated. So it helps in translation. Next is splicing. So splicing, it cannot ensure splicing is also there. Next is alternative splicing. Do not ensure alternative splicing. Where this intron exome, it do cut it in an alternative manner, right? And alternative splicing also require a lot of enzymes, which is a part of your post-transcriptional modification. And this alternative splicing is a part of post-transcriptional modification. So it, and capping is done after that. Okay. Okay. So here this uh, overall explains why this alternative splicing is the answer. Yes, first question. Fine. So first question, which RNA is abundant in human cell? Here I am asking you which is the highest percentage amount in RNA. So mRNA, tRNA and rRNA, if you see here, mRNA has only 5 to 10% of total RNA. tRNA only 10 to 20% of total RNA, but rRNA 60 to 80% as it makes the ribosomes. Okay, ribosomes are required for translation process and rRNA make the ribosomes are made of ribonucleic acids. That's why it just makes the highest abundant of RNA. If the question is which one is the longest RNA, so longest RNA will be mRNA because it is just a long chain. Understood? So if we just modify the question in a way like that, longest RNA, largest RNA, or abundant in RNA, so the answer will be different. Here I am asking you which one is the highly abundant, means high percentage is already present inside the cell. So that's why your answer will be rRNA. Okay? Okay then. Thank you. Okay then, I guess it is clear for everyone. Thank you for joining. We'll see you on next Monday. Okay?